Hi everyone, this is Sarah Dean, and I want to talk about white blood cells today and how to identify them under a microscope. Before we start in on that, I'm just going to give a brief introduction to whole blood. So I'll just write blood up here since that's our topic for the day. So if you were to take a blood sample from a patient and then centrifuge it, so put it in a centrifuge and it gets spun really, really fast, and centrifugal force pushes the densest, heaviest portions of the blood to the bottom of your little vial, and the lightest or least dense portions are at the top of the vial. This is what it would look like, and this is a good tool. A centrifuge is a good tool that you can use to look at someone's, just get a rough look at someone's blood content. So here's the blood sample. So this whole thing from top to bottom would be whole blood, but we've separated the parts now in the centrifuge. So that's whole blood. Now at the very bottom, the densest part of blood is the hematocrit. And so that's going to be almost half. That's going to be red because the hematocrit is the red blood cells. So let's see here. Color that in. Right. So this is the hematocrit. That's about 45% of the blood volume. This is your red blood cells, also known as, the more scientific term would be erythrocyte. Right. Then just above that, we have this sort of milky looking layer. And let's see, that's hard to see. Let me make that a little darker here. Let's do this. I'm just going to make it yellow, but the truth is it looks white. And we call this the Buffy Coat. Still really can't see that, so. Okay, so this kind of milky layer we call the Buffy Coat. That's going to be less than 1% of the total blood volume. And this is going to contain your white blood cells and your platelets. So white blood cells, also known as leukocytes, that's the more jargony term for that, leukocyte. So whenever you see the term cyte, C-Y-T-E, we're talking about cells. Leukocyte, erythrocyte, it's a term for cells. And leuko, that refers to white. So the word for white is leuc or leuko. So white blood cell, leukocyte. We also have platelets. Platelets are really important for blood clotting, like when you get a scab. They're tiny cell fragments. So platelets, the jargony term for that is thrombocyte. And then at the top up here, the rest is just what we call plasma. And this is going to be about 55% of blood volume. Right. So these values can vary even in normal healthy people and across men and women. But if they vary, if somebody's hematocrit is unusually low or unusually high, it can mean, various unhealthy things like dehydration or problems like that. Okay, so if we were to just look at these together, the Buffy Coat and the hematocrit are known as the formed elements of blood. They're the living component of blood. But even when looking at formed elements, most of those formed elements aren't actually cells and are not truly living. So for example, red blood cells have no nucleus. They have no DNA anymore. So they're basically just bags of hemoglobin, and they don't last very long because of that. They can't regenerate. So they are formed from true cells in the bone marrow, but they themselves are no longer true cells. And it's the same for platelets. Platelets are cell fragments. There's, in the bone marrow, you have this larger cell called a megakaryocyte, and it's this huge cell with a multi-lobed nucleus because the DNA in the megakaryocyte replicates, so it undergoes mitosis, but without cytokinesis. So the cell does not divide. So it can have these huge nuclei that are multi-lobed that haven't fully separated from each other. And it's this is the megakaryocyte. So like I said, this is in the bone marrow. And it reaches, so this huge cell with multiple lobed nuclei. It looks like it has many nuclei, right? Because it kept replicating its DNA without dividing. And it has these long branching arms that extend out into the capillary beds, and they break off, sort of like leaves in the autumn break off from a tree. So they break off into fragments, and these are platelets. So the platelets just have a little cytoplasm in them, and they're surrounded by the cell membrane, but they are not a true cell. It's just a cell fragment. So platelets, red blood cells or erythrocytes, and white blood cells or leukocytes. These are all the formed elements, but only these are true living cells. These are really just cell fragments or the remnants of a cell, not true cells. All right, so we're now going to zoom in on the Buffy Coat and talk about the Buffy Coat specifically. So let's go. I have some images from Google image searches to help discuss this further. So here's all of the formed elements from blood. Oh, and I want to add, actually, back here, that blood is considered a connective tissue. Sorry, before we move on. Sort of like cartilage. It's strange, isn't it? It doesn't seem much like a connective tissue. It's very unlike the others. But a connective tissue has to have a network of fibers and a ground substance, and then a living component, a living cellular component. So in the connective tissue, the plasma is the non-living matrix. The liquid in the plasma and all the dissolved components are the ground substance, but there actually are fibers in the blood, but they're dissolved. So you have these proteins. There are certain proteins that are dissolved, and when they get the signal to clot because you have a wound, they will form these branching networks of a protein called fibrin. So fibrin forms fibers when given the signal. So usually these proteins are just dissolved in the blood, and so we don't see very easily that there is this fibrous component like in other connective tissues, but it is actually there. It's just dissolved, but when needed, it will form that branching network that's necessary to form blood clots. And then, of course, there's the living component of the cells. All right, so let's go back to this image. So these are the red blood cells, and we're not going to focus on those. And these platelets or thrombocytes, we're not going to focus on those either. So we already talked about these are not true cells. They're still very important. The red blood cells are just sacs of hemoglobin, and hemoglobin is a protein that binds to oxygen and carbon dioxide, and it allows the red blood cells to transport those gases through the body and deliver them to where they need to be. The platelets, again, important in blood clotting to stop bleeding at a wound site. Now, the rest of these are white blood cells: basophils, neutrophils, monocytes, eosinophils, and lymphocytes. So those are all white blood cells, and you're going to need to learn how to identify them under a microscope in real life, and you're going to need to know what they each do for our immune system. So a lymphocyte, we can divide these into granulocytes and agranulocytes. So the granulocytes, these are the basophils, neutrophils, and the eosinophils. Granulocyte means that these cells look granulally under a microscope, and you can see that in this cartoon image. You can see these basophils have these blue granules. The eosinophils have red granules. And the neutrophils, their granules are very fine, so they almost just look like a dusting. But, and in here they look kind of lavender. They're actually a mix of red and blue, and so red and blue together make purple or lavender. So red and blue granules. These granules contain antimicrobial compounds, because leukocytes, or white blood cells, they're designed, they help you with immunity. They fight infection. So granules with, oops, oh no, I switched, we'll get to that image later, with antimicrobial compounds to fight infection. Now, so these granules contain all of these like weapons that those white blood cells can use, those granulocytes can use they exocytose the contents of those granules onto the microbes to fight infection. Now, agranulocytes, these are going to be the monocytes and the lymphocytes. All right, so that's this one and this one. These are called agranulocytes because you cannot see the granules. They actually do have granules as well that also contain antimicrobial compounds, but they don't pick up stain. They don't pick up the stain we use to visualize these cells under a microscope, so you don't see the granules. So we'll say, so agranulocyte means no granules. It's not that they don't have granules though in real life, it's just that they're invisible. All right. So monocytes, once activated, can become different cell types. They can become dendrocytes, and macrophages. Sorry, my allergies, this juniper is just destroying me. There are two different kinds of lymphocytes, and those are B cells and T cells, which you may have heard of before. The next few slides we'll talk about more about how to identify them and their function. But right now, the last thing I want to put out here is their relative abundance. So we already talked about how the Buffy Coat, which includes the platelets, so the platelets are in the Buffy Coat, the lymphocytes, the eosinophils, the monocytes, the neutrophils, the basophils. So all the white blood cells and the platelets are in the Buffy Coat, which makes up less than 1% of whole blood. Now, there's still different abundances of each of these, so some are more abundant than others when you're just looking amongst the white blood cells themselves. So the best way to remember their relative abundance is the acronym, or the phrase, never let monkeys eat bananas. I mean, of course you should let monkeys eat bananas, but this phrase helps remember the relative abundance. So this is the highest abundance here and the lowest abundance on the right. So never, the N stands for neutrophil. The L stands for leukocyte. Or, I'm sorry, lymphocyte. And see, I'm running out of space here. Go down here. Lymphocyte. M stands for monocyte. E stands for eosinophil. B stands for basophil. And so there are normal ranges of abundance. You know what, I'm just simply going to erase the red and blue granule comment. Oops. Because I don't have space for it anymore. Maybe I'll put it up. Oh. Technical difficulties. Let's get more carefully. All right, so back to black. Amy Winehouse song. Okay, so red and blue granules. Okay, so neutrophils, there's a normal range for each of these white blood cells, and if you do a white blood cell count and they're out of their normal range, it can indicate various styles of illness or infection. So neutrophils, they make up about 50 to 70% of all the white blood cells. So obviously this isn't 50 to 70% of the whole blood, because all of these white blood cells together make up less than 1% of whole blood. So it's 50 to 70% of the total white blood cell count. Lymphocytes are 25 to 45% of all the white blood cells. Monocytes are 3 to 8%. Eosinophils are 2 to 4%. And basophils are less than 1% of the total white blood cell count. So basophils are very rare. And I'll make a comment about the colors here. So these cells don't have any color naturally. The colors are created by the dyes we use to stain them to make them stand out more under a microscope. And we have acidic dyes and we have basic dyes. So eosinophils are acid lovers. They're acid lovers, meaning they absorb the acid dye. They themselves are basic, which is why they like the acids. The acid dyes stick to them. So eosinophils like the acid dye. Basophils stand for basic. They like the basic dye. The basic dyes stick to them. Neutrophils, neutro meaning neutral, they're in between acidic and basic, and so both dyes stick lightly to them. And so that accounts for the different colors. The acid dye is pink. The the basic dye is blue, and then the neutrophils pick up both of those, giving them a lavender look. All right, so let's go on to the next image here. So here's a microscopy image of these cells, and so you can see what they look like in real life. And we'll look at a couple of these. But these are the red blood cells. And these are good to know because one way you can identify one white blood cell from another is looking at their size relative to the red blood cells, especially in pictures, because in pictures, you're dealing with different magnifications. And so a particular white blood cell may look big or small depending on how much it's magnified, obviously. So the best way to figure out how big it really is to look at the red blood cells next to it. It's always going to be surrounded by red blood cells because red blood cells are far more abundant in the blood than the white blood cells are. So these are the red blood cells, or the erythrocytes, and you can see that the platelets are very tiny. The platelets don't always cluster like that. Sometimes they just look like a fleck. Usually they just look like a fleck here and there, mixed in with the red blood cells. So they don't have to cluster into a pack like that. They don't always. All right, so here you have eosinophils. So I'm going to talk a little bit. We have already talked about their color, so now I want to talk about their size and some other features about them. So eosinophils are about twice the size of red blood cells, maybe a little bit bigger. So approximately 2 times red blood cell size. Neutrophils are also about 2 times the size of red blood cells. And basophils are, so basophils are a little bit smaller, but you could still kind of say, you can see that there's differences in sizes amongst these, so basophils are kind of smaller than the other two. But if you look at the average size across basophils, they're still about twice the size of red blood cells. Monocytes, on the other hand, are huge. So first I'll say the 3 granulocytes that we talked about, the 3 granulocytes, are about twice the size of red blood cells. They're all approximately the same size as each other. Monocytes are much bigger typically. They're more about like 3 times the size of the red blood cells around them. And lymphocytes are very small typically. Usually lymphocytes are about the same size as red blood cells, so equal the size of red blood cells. So the agranulocytes have the smallest of the white blood cells and also the largest of the white blood cells. So the smallest is approximately the same size as red blood cells. The largest is approximately 3 times that of a red blood cell. And then the 3 granulocytes are about the same size. So size is one way you can start identifying which kind of white blood cell you're looking at. So how to ID white blood cells. Okay, so first thing is size, particularly relative to the red blood cells. The next thing you want to look at is the color, which we already talked about. So you want to look at, are there granules or not? And you also, so granulocyte or not, and then color of the granules. All right, and then, so if we look back at these, the neutrophils have very, very fine granules, and they're lavender, right? So lavender granules, we looked at this in the last slide. The eosinophils have coarse granules. They're bigger. You can really see those granules, and they're red. And then the basophils have the biggest granules. Their granules are so big and so dark blue that sometimes you can't even see the nucleus. So they have dark blue, huge, coarse granules. So smallest granules in the neutrophil, medium granules in the eosinophil, biggest granules in the basophil. Lavender for the neutrophil because it's picking up both the acidic and the basic dyes, both the blue and the red dyes, which makes purple. Eosinophils pick up the acidic red dye, and the basophils take up the basic blue dye. And then you have the agranulocytes, the monocytes and the lymphocytes. And then you want to look at the shape of the nucleus. All right. So the shape of the nucleus, you have, so in neutrophils have these many lobed nuclei. They can have 3 to 6 lobes. So if you look at this neutrophil here, like there's a lobe, this is like maybe 2 lobes, maybe all one together. Okay, so they have these like very lobe shapes. So the neutrophil will say 3 to 6 lobes for the nuclei. Eosinophils, E for earmuffs. So they're bilobed. They have 2 lobes, and those lobes look like earmuffs. So bilobed. And I like to think of E for earmuffs. E for eosinophil, E for earmuffs. Oops. All right. The basophils are usually bilobed as well. But like I said, it's very difficult to see the nucleus most of the time because those granules stain so dark and the granules are so big. So this is also all right, but obscured. Monocytes, they have not, their nuclei are not usually fully lobed. So they have a strange shape, but the different compartments of the nucleus are not so distinct from each other. They look more like a kidney bean. So they got this like this weird bent kidney bean shape. And then the lymphocytes, not only is the nucleus a normal round shape, but it takes up most of the cell. Nucleus round takes up most of cell. So the cytoplasm around the nucleus in lymphocytes looks like a crescent. Okay, so lymphocytes I think are the easiest to identify. They're really not much bigger than a red blood cell, so they kind of fit right in with the red blood cells around it except for the color. The color is dark purple with like a light purple crescent with a dark purple center. The monocytes have a light purple cytoplasm as well with a kidney bean center, and they're huge, so those are also fairly easy to identify. So you're mostly going to be struggling with the granulocytes. So the granulocytes, neutrophils are the next easiest to identify because they have those wonky, like many lobes, 3 to 6 lobes. Eosinophils and basophils, they're kind of the same size. They have the same number of lobes for the nucleus, but luckily they're distinctly different in color with the stains that are used. So eosinophils are red because they pick up the red acidic stain, and the basophils are blue because they pick up the blue basic stain. So you have to take into account all of these things, to the size, the color, and the shape of the nucleus to determine which white blood cell you're looking at. If you're looking at a slide, you can also kind of try, if you know that the slide is a blood sample from a normal, healthy human, you can also look at relative abundance and see how rare they are, because basophils are super rare. They're really hard to find on a slide of a human blood sample. All right, so let's look at the next picture here. Oh yes, this one really shows you these platelets better. So those platelets, they're not always lumped together like in the last picture. They're kind of just flecks throughout the slides. And then this, again, so now what is this? So if you had a test question, what is the cell? So remember, we looked at our 3 criteria that we have to look at: the size, the color, and the shape of the nucleus. So the size, it's huge. So size, huge. That tells you it's probably a monocyte. But we can continue forward. So color, does it have granules or not? I'm including that into this. There's no granules. No granules. It's agranulocyte, so that fits with the monocyte hypothesis. We don't see any granules. And then nucleus, kidney bean shape. So all of those 3 characteristics tell us that this is a monocyte that we're looking at here, which I said in the first slide can they can divide and become either dendrocytes or macrophages, which have slightly different roles in immunity. All right, here's our last slide to look at. And of course, it's really important to look at many different slides when you're studying, because these cells, you're looking at them from different angles and different pictures. The dye might have adhered to the cells slightly differently in different slides. The slides might be different ages, different magnifications, and they're all going to be a little bit different. They're just going to be different. So you want to get a sense of the range of ways that each of these white blood cells can look. So in this slide, I'm going to talk about their function as well. So here's this neutrophil. You can see all these different neutrophils. But one thing that you can really see that's distinctive in all of these pictures is that there are many lobes to the nucleus. The nucleus is that dark thing in the center, and instead of just having a round, simple nucleus like you've learned about in the past, it's got this really wonky shape with all these different lobes. So this is definitely a neutrophil just on that criteria alone. There's 3 to 6 lobes in these pictures. So neutrophils, what are their function? These are phagocytes. Now this is a gross oversimplification of what they can do, but that's their most important function in immunity. They are phagocytes, which means they phagocytize or ingest and digest pathogens. And in order to ingest something, it has to be smaller than you. So this is where your prior knowledge of eukaryotic cells and prokaryotic cells come in handy. You know that your cells, including your white blood cells, are all eukaryotic. So those are pretty large cells. Bacteria are far smaller. So a neutrophil can ingest and digest bacteria. Viruses are even smaller than that, so it can also ingest and digest viruses. It could ingest and digest toxins released by pathogens. But it's not going to, it's very unlikely to ingest another eukaryotic cell, so a eukaryotic invader. So eukaryotic invaders to your body, eukaryotic pathogens, are going to be things like fungus. Fungi are eukaryotic. That means that fungal cells are the same size as white blood cells, so how's it going to eat it? That would be like you trying to gulp another person down in one bite. It's just not going to happen. So it needs to be, and also protists. So protists, like malaria, those are also eukaryotic cells, so they're very, very large. So it's very unlikely for neutrophils to ingest and digest, to phagocytize any pathogen that's eukaryotic. So they're really focused on bacteria and viruses. All right. Let's see. I want to stick with the granulocytes for now. So let's talk about eosinophils. And I'll do this down here. I'm going to actually, so eosinophils. All right, so you can see that they've got that red look to them because they absorb the acidic red dye, and they've got that, for the most part, that earmuff look. Now, you can see they're not always the same. Like, this one's kind of weird. It would confuse you because it looks like it has 3 lobes, and maybe that's just how that particular cell is positioned in the slide. This is why you need to look at more than one feature in case you have a sort of unusual angle on a cell, or it's just a little bit weird. It's not the typical version of an eosinophil. So you really want to be able to look at all those things: the size, the color, granules or no granules, and the shape of the nucleus. So eosinophils have that earmuff shape to the nucleus, typically speaking, and also, very importantly, they pick up the red dye. So the most important thing about eosinophils is they kill the larger parasites. So we were just saying neutrophils can only phagocytize small parasites. Eosinophils are going to take larger parasites. And they're not going to endocytose them, or phagocytize them, because they can't. They're the same size as these parasites. So what they do is they excrete, they exocytose those the contents of those granules. So they sidle up next to a parasitic worm or a fungus, and they just exocytose all of those antimicrobial compounds onto the invader. And what's inside those granules for the eosinophil are digestive enzymes. So they're actually digesting the parasitic worm or the fungus or the protist outside of the cell. So the neutrophil is going to bring the small parasites in and digest them. The eosinophil just like vomits out its digestive enzymes and digests it from the outside. So it's going to take, like parasitic worms, like I said, fungi, things like protists. So these larger invaders that you don't want in your body. They also are known to play a role in allergies, but that's when they go wrong, right? Allergies are your immune system gone awry. When you get allergies like I have today, I'm having an immune response to harmless things in my environment. That pollen is not going to invade my body. It's not going to eat me up. The only reason the pollen's giving me a problem is because my immune system is incorrectly reacting to that pollen and treating it like an invader. So eosinophil reactions gone awry can cause allergies. Wrongfully activated. So when wrongfully activated, they can create an allergic response. There's no good reason for allergies. It's just your immune system messing up, basically. All right, now let's do basophils. All right, those are at the bottom there. So again, you can't even really see the nucleus because those granules are so dark and so large. So that is a very good way to tell a basophil. And they release histamine from their granules. So histamine, release histamine. Histamine is an inflammatory. So what it does is it causes blood vessels to dilate and all the blood rushes to the area, causing inflammation and a burning, warm sensation. Now, we don't like inflammation. It's uncomfortable, but it's actually a really important part of your immune response. It helps bring oxygen and other white blood cells to the site of infection. It brings platelets and clotting factors to contain the infection. So your inflammatory response is actually good. It helps you fight infection, unless it goes awry. Like over-inflammation can cause problems. If you have an allergic response that's no good, no use to you, you might want to take an anti-inflammatory and antihistamine. So antihistamines block the action of histamine. They block basophil action. So again, when these basophils are wrongfully activated, you get an allergic response. So that's true for eosinophils and that's true for basophils. When either of them is wrongly activated, you get an allergic response. So they're responsible for inflammation, which is uncomfortable but very important for fighting disease and infection. All right, so now let's move on. So those are all the granulocytes. Now let's move on to the agranulocytes. I want to talk about the lymphocytes first. The main function of lymphocytes is immunity. Immunity. That's immune system memory. That's why you get vaccines. So when you get a vaccine and it gives your body it teaches your body about a pathogen so that the pathogen can no longer successfully invade your body and make you sick. What you're doing is you're training your lymphocytes. The vaccine activates your lymphocytes. It teaches them or trains them about the pathogen. So if you get a chickenpox vaccine, you're taking little proteins that are found on the surface of the pathogen that causes chickenpox, and you're introducing it to your B and T cells so that they recognize it and they know what to look for. That way, if they ever encounter real chickenpox, they will nip the infection in the bud before it has time to get into your cells and cause disease. So now we've got the COVID vaccine that people want. That's the same idea. You've got this vaccine that has proteins that you find on the surface of coronavirus from 2019, COVID-19, and so you introduce those proteins from COVID into your B and T cells. It activates them, create an army of those T and B cells that are designed to attack coronavirus. Now, if you ever encounter coronavirus, if it ever gets into your body, they're going to destroy it right away before it has time to replicate, before it has time to start disease. So immunity, that's immune system memory. So that's what your lymphocytes are for. Now, finally, we have monocytes. And we've already talked about how these divide into macrophages and dendrocytes. And these are phagocytes as well. That's their main role. But they're a smart kind of phagocyte. What they do is, after they ingest and digest a pathogen, they take pieces of that digested pathogen and show it to the B and T cells. So they communicate with the lymphocytes to amplify lymphocyte memory. So they introduce, so the macrophages and dendrocytes can go around, digest pathogens, and then present pieces of those pathogens to the lymphocytes to teach the lymphocytes. So they communicate with and teach lymphocytes. Communicate with lymphocytes about their kill. So those are the main functions. I know there's something called bands here. Just ignore bands. We don't need this in this particular slide. These are the things that you need to know for this class. So neutrophils, lymphocytes, monocytes, eosinophils, basophils. These are the agranulocytes. And they're both important for immune system memory, as it turns out. So that's another way you can classify them. Important for developing immunity. And then these are the granulocytes. They don't develop long-term immunity, but they help you fight a current infection right now really quickly. So that's their job in different ways. They phagocytize or digest outside the cell, or release histamine and cause inflammation. And those are your white blood cells.